Our next presenters are Dr. Maureen Fitch of the Hawaii Agriculture Research Center and Dr. Mike Melzer of the UH-CTAHR Plant and Environmental Protection Sciences. The presentation is entitled Propagation of Reduced Virus Green Tea Plants for Hawaii Growers. Dr. Fitch is a plant physiologist at the Hawaii Agriculture Research Center, also called HARC. Her group does clonal propagation of papaya, some anthuriums, and other plants as requested by local growers. They also assist growers with concerns about the identity of their papaya cultivars. Having been on the team to develop the world's first genetically engineered virus-resistant papayas that have helped save an industry, they continue to assist that and other industries. Recently, they shipped 700 plants of cloned rainbow hermaphrodite papaya plants to Hawaii Island to a grower who was impacted by the 2018 eruption and as support for losses incurred and to experience a new planting method. In the new project, cloning Dr. Mike Melzer's reduced virus green tea leaf plant clone, they will assist local growers in the lau trade, ornamentals, and other businesses by supplying stock plants as replacements for their virus-infected plants. Now, Dr. Mike Melzer is an associate researcher at the University of Hawaii at Manoa who specializes in agricultural biosecurity. In partnership with state and federal partners, his program conducts statewide surveys for exotic pests and pathogens of regulatory importance. As the Hawaii representative of the National Plant Diagnostic Network, his USD-accredited laboratory develops and utilizes diagnostic tools for plant pathogen detection with an emphasis on vegetatively propagated crops of importance to Hawaii. With support from the National Clean Plant Network, his program generates, maintains, and distributes clean germplasm to nurseries and growers in Hawaii. His program also administers the multi-agency response to the coconut rhinoceros beetle invasion on Oahu and works to develop management tools for this pest. Originally from Canada, Michael received his bachelor's degree from Trent University before receiving both his master's and doctoral degrees from the University of Hawaii at Manoa. Dr. Melzer, welcome.
Thank you. Thank you for the introduction, and thank you for the opportunity to present to the association. I very much appreciate it. I am going to share my screen. Today we are going to discuss clean tea plants. And we say clean, we're not talking about, they're dirty, they got some dirt on them, where we're basically talking about systemic pathogens, and typically we're going to be talking about viruses, something that's obviously in the news a lot these days. So really what we're discussing is, propagation of these reduced virus green tea plants for the Hawaii growers. And for those of you interested in contacting me, you can see my contact information through phone, email, or our website. So let's get started. I don't think we need a great introduction on this plant. I think everyone's familiar. Let's see, there we go. The tea plant, tea in Hawaiian, Cordyline fruticosa. Basically, this is one of the canoe plants. And the common green variety was initially introduced to Hawaii by Polynesian settlers. Interestingly, throughout the Pacific, this green tea is actually a sterile plant. We do see it flower and occasional make fruit, but typically, 95% of the pollen is sterile, and so they will flower, but generally, this is only going to be propagated through cuttings. And that becomes important, as we'll discuss later, when we're propagating plants strictly by cuttings instead of letting it go through seed. So after this initial introduction, obviously we've had many subsequent introductions of tea, different varieties. These are the ones that are typically being bred in ornamental breeding programs, and we have a lot of both professional and amateur tea leaf breeders in there. These are basically grown as landscape ornamentals or for cut flower arrangements. And conversely, these ones do flower and generally produce viable seeds. So you can propagate from these plants, and that gives them some sort of advantage. Obviously, that's important for any breeding that's going on, but also that going through the seed stage is going to help eliminate some of the virus load that the parental plants might have had. Typically, viruses and systemic pathogens will not be spread through seeds. They basically get excluded. And that can sometimes contribute to hybrid vigor, but it's that's one of the components. But it's this isn't necessarily the case. There are some seed transmitted viruses, but all the ones we're going to talk about today are not seed transmitted. So I was introduced to this plant by a colleague, a fellow graduate student. Her family had a tea farm up in Kahuku, and I spent a few weekends helping them plant it. As long as I can figure out which end was up when we stuck the canes in the ground, I think I did a reasonably good job. So I had fairly good experience with this. But when we look at back to what tea is used for, there's obviously a lot of traditional uses through thatching, through houses. We've got the rain cape or the ahula'i that was used, traditionally thatched from tea leaf. And we can't discount okolehau. That's basically a distillate from fermenting the corms. The corms of these plants are large, and they have a reasonably good sugar content, which allows them to be fermented. And they also did serve as famine food when, say, other crops, they collapsed either for whatever reason. Other preferred root crops such as taro or sweet potato, the corm is edible. Today, I think we see tea used quite a bit still, maybe not in their traditional means. Although I think there are some people making okolehau, but I'm not sure if it's actually coming from tea plants or so forth, or whether it's being distilled from other grains. But we see tea leaf when we look at the Merry Monarch, when we get our lau lau, and also, well, thank you. This isn't tea, but also for lei, and thank you very much for the lovely lei that was sent. Very much appreciated. So they are used still in the cut flower industry and as lei. So a little bit about commercial tea production. Everyone's got probably a couple in their backyard, but there still is a commercial industry here, most of which is sold to the food industry for lau lau, and I think Maureen will give some details on that. But some of the higher quality leaves, they can be sold for ornamental or cultural purposes. And sometimes that's the more desired because they're higher value. When the hula halau come through, they can harvest leaves from areas. And if it's from a commercial farm, those leaves typically have a higher value. The sort of sad news is that, in the recent years, and the most recent information I could find was 2013 to 2017, the number of growers are going down. And if you look 10 years ago, that number was in the 40s. And so now we're in probably the mid to low 30s for the number of commercial tea growers here in Hawaii. And one of the casualties was my friend up on the North Shore. Her farm is no longer there. But right now we're selling between 1.6 to 2 million, almost 3 million leaves per year. And these are sales, modest, but still important, between 150 to almost $250,000 in sales. And to go back, 10 years ago, that was probably over $500,000 on good years. So unfortunately, it is an industry in a bit of decline right now. But the current growers are still vibrant. So I got professionally involved in this plant about 10 years ago. And this was with the advent of this disease called tea ring spot. And this is, some of you might know, a retired extension agent, Ed Marcino. He's the one that brought these samples to our lab. This was when I was doing my PhD at the time. And he brought these in, and he said nobody had seen these symptoms before, these ring spots. And we can see on the leaf here how they would be, pretty small, about they were basically interveinal. They basically showed up in the veins, but they could coalesce into larger spots. And he was describing how on the windward side of Oahu that this disease was just spreading rapidly. It would show up in one corner of the field, at the beginning at one season, and by the end of the season, the whole field was infested. And we've done subsequent surveys, and basically it's a statewide issue now. So this had, initially had an issue for the Lau folks. They didn't like blemishes on the leaves, although I think now they sometimes do take them. But definitely, even though I find the symptoms kind of pretty, when you're using this leaf as some sort of in an ornamental setting, as a cut flower or as a lei. However, I have seen lei made with these. I actually, a graduate student worked on this, and I found a lei at one of the lei shops with tea, and it had these symptoms on it. So I had to get that one for him when he gave his master's degree, and he actually worked on this virus. But anyways, yeah, so we thought this was a viral disease that's affecting tea. And this was sort of significant because really there hadn't been much work done on viruses infecting tea. In fact, the only report of a pathogen infecting tea is tomato spotted wilt virus. And we basically, that was discovered here in Hawaii, and we basically discounted that was not responsible for this. So, over a few years, we did some work on this. We did a technique which was actually pretty new back then. 10 years ago, high throughput sequencing was not common. Now it's a very common technique. And using this approach, basically we can find out anything, all the DNA, all the story that sample has, anything about its DNA or any pathogens it has, including viruses. And wow, did we find viruses? We couldn't even be creative in naming these things. We just called them Cordyline virus 1, 2, 3, and 4. And this is just the ones, we had to stop somewhere. So we stopped at 4. These were all sort of related ones, and we found, half a dozen other ones in there. And so this is what happens when you've got, as I mentioned, a plant that is vegetatively propagated and not going through seed, that it's accumulating all the viruses through its history. So these green tea plants that were brought in by Polynesians, they have been accumulating viruses the whole time because they haven't gone through seed. We did find finally, and we actually just published this a couple of years ago, we finally found the causal agent. It's a new virus we called it T ring spot associated virus. It's spread by an aerophyid mite, really tiny mite. And that's how it spread so quickly through fields. Those things are spread by the wind, so it's just like dust blowing through a field. So we sort of knew what that one did, but we found these viruses have a huge viral load. Think of us, they can't cure themselves from viruses. So think of us if we got the cold, the flu, we got other different viral diseases, and imagine we didn't have our immune system and we were just fighting those for our entire life. And so the question sort of becomes is, what do all these viruses that are sublethal, they're not killing the plant, but surely they must be, somewhat slowing it down. And so to explore that, we decided, and this was just sort of a side project. I really never thought it would go anywhere. But I had a lab at my disposal doing my graduate work, and why not take advantage? So I selected just an average looking tea plant growing in Wailupe Valley here in Oahu while I was hiking. Nothing spectacular. It wasn't healthy, but it wasn't unhealthy. And I said, let's see what happens if we eliminate all the viruses in this tea plant. Let's see what happens. So we brought it into tissue culture, propagated it up, and we did a technique called cryotherapy, which is basically we're just freezing. Well, we basically treat the plant with some chemicals, basically antifreeze if you want, and we take it through this technique called cryotherapy. And what happens with cryotherapy is that it kills all the large differentiated cells that have these big water-filled vacuoles. When they freeze, they just like water, it expands, it turns to ice, and it kills the cell, it breaks open the cell. But those small, and if you look at this picture here, this is an example of a virus-infected plant, the meristem, and we can see the purple is where all the virus is, but we can see in this meristematic tissue, this replicating tissue, these cells are virus-free. And so basically what cryotherapy is doing is killing all the differentiated cells and just leaving these replicating, these small meristematic cells. And so we basically regenerate these plants, and we can see here, maybe you only get about 10% surviving after they've been frozen. They basically go into liquid nitrogen, and we can see a couple that are surviving here. And we would just sort of grow them up And I had, and then this is just something we put them in tissue culture, we continue them in tissue culture, regenerate them from just a few cells, those few meristematic cells. And then we tested them, and sure enough, we cured all the viruses that we knew about that was in that original plant from Wailupe Valley. And so, yeah, this sat in our, I never really thought about it all that much, but then the plants were getting big. So I was like, well, I might as well plant these things out. And I put them in one of our greenhouses up at Pope and just sort of ignored them for a while. And then it came to a point I couldn't ignore them anymore. This is what happened after they got into the greenhouse. They kept breaking open all the pots we were growing them in. And, I was just, when they first grew up, they looked fairly normal. When you see these basil leaves here, they just look like normal tea leaves, but the plant just kept growing and growing and producing really large, blemish-free leaves, basically. And so I was just really impressed. But again, this is greenhouse production. They don't have the wind damage in that, but you could not find a single spot on any of these leaves. And this, I would say this doesn't look like the average tea plant that's growing around in people's backyards. So that's been sitting there for 10 years, and then it's just been in the last couple of years, and I'm going to let Maureen take over this story from there. That there's started to become a little bit of interest in these plants. So I'm going to stop it there, but I want to acknowledge all the folks that help with this. I think my 15 minutes are up, but a lot of folks in my department, Ed Marcino, Jari Sugano, who actually got us that official initial grant to do some of this work from Department of Ag, other folks in our lab, and Myron and Maureen for their persistence in moving forward with this project, and then National Clean Plant Network, because we're basically applying the principles of that network to this project. So with that said, I'm going to turn it over to Maureen.
Because I've been around in Hawaii for a long time, playing around with plants at heart, and my friend Myron Murakami, his wife Carol, comes to the lab once a week to help us. And in May, Myron called me. He said, "Hey, Maureen, are you looking for a new project?" I said, "No." "What?" "Because no matter what Myron asks for, Maureen has to say yes." So he says, he tells me about this green tea, and I didn't know about all the viruses. And I've met Mike Melzer once at a taro field day about 3 years ago. And so he told me about this cryopreserved green tea that's clean. And he says, it's really good. It's growing at my friend Scott Chun's place, and it's amazing. So I said, okay, well, we need money, so started thinking about grant proposals, and I was given plants by Myron and talked to Mike, and Mike gave us some things to start with. So that's how I got into this business, okay. Like Mike said, Hawaiian green tea is a canoe plant. It came in 700 AD with the Hawaiians, so it's been around a long time. One of the reasons the growers like it is because it doesn't flower, not that much. And every time a plant flowers, the leaves get smaller and it spends all its energy making flowers. So they don't like the flowers. Myron and others harvest tea leaves twice a week for laulau, the laulau industry. They sell their leaves to keokis, and I think you've seen these laulau in all the supermarkets, at Costco. They're even exported to Gardena and Seattle, LA, because of all the locals who live in these places. They're used in flower arrangements. I didn't see any in the flower talks earlier, but I'm sure they think about using them. And they're good luck. We wave them at football games. Also, they're important, like Mike said, in tea leaf lays. And I guess if they have a few viruses, it's okay. Because tea has been here for centuries, lots of chance to get infected by things. And we are here now and we have become infected by COVID-19. Probably first came here with travelers, both of them, and other things coming in, like recently there's the coffee rust. Some viruses damage the leaves so badly that businesses like the tea leaf growers who sell to lau makers, they can't supply their customers sufficiently. And it's a lot of work to find really nice leaves. Mike has said that tea has 6 or 7 viruses. They know of 5, and his group had identified 5. This slide shows tea leaf plants from Myron and Carol Murakami's current field. You can see the streaks, and you can see that there are not many leaves on their plants. And it's really a stretch for them to twice a week go into their field and collect enough leaves to take to keokis. The middle plant is something from Scott Chun's. Scott Chun was very sharp about 2 years ago. I guess he met Mike, and Mike said, oh, you can have some plant material. So Scott took him up on it. Myron said, I should have been more sharp, but he didn't. Now he wants to play catch up. So this is a plant at Scott Chun's field. It's beautiful. It would make a nice potted plant. And this is Carol and Sarah Murakami standing in front of some 2-year-old plants that Scott had originally gotten from Mike. And they've grown up and they've been cut several times. And you can see how bushy Scott's stand is compared to Myron's. Okay, so Mike's cleanup method that got rid of at least 5 viruses is pretty cool. And after Myron, the fellow in the blue shirt, went to Scott's with the hat, Myron decided, I want some of this stuff. And so he approached me. And so we are trying to figure out how to grow more plants. For Lau Lau, this is my husband, Ron, holding a yardstick. This is 19 inches here, okay. For Myron, he can't find a 19-inch leaf. It's really hard. Scott has 25-inch leaves, no problem. The leaves from his young plants are long enough, and they can harvest many, many, many. I was looking at this leaf though, and I want Mike to look carefully at it because I hope those aren't viruses. I never noticed it when we're taking the pictures, but sun was coming through and I don't know what they are. Hope they're not viruses. Anyway, this is Myron showing how after harvesting maybe 4 plants, still there are about 6 of them left, and they rotate. So they have a lot more leaves coming out. Everything on the plant is beautiful. There's not one spot. And Mike, you should be very, very proud of your children. So HARC will try to provide plants to growers. We can take the lateral shoots from the field plants and pot them in 4-inch pots. Whatever we provide to the growers, we'll have them in little pots because then we can shove more on our greenhouse benches at HARC in Kunia. We will also cut up some rooted cane pieces, some canes, and root them, put them in 4-inch pots, and then growers can have them. And finally, we will micropropagate small plants, grow them up into 4-inch pots. And so that's what we will give these growers. And 7 of us have given us some funding as seed money. So they're the ones who'll get the plants first. These are Scott's 2-year-old plants that have been cut many, many times. And this is one of his production fields, but he's got 2 others. Anyway, you can see where you can pull off lateral shoots and root them, and maybe they're a little large for 4-inch pots, but we'll try. These are cane pieces that Myron has been growing for about a month. He cut them up, put them in 4-inch pots, and they started making shoots, at least 2 per. When they're skinnier, they make 1, and that's kind of how we can do it at HARC. And finally, we plan to micropropagate and grow the plants large in the greenhouse. And we've been fooling around with different media, stuff that we had in the cold room, and almost everything worked. So we're off to a good start. We're still looking for grant money. we'll get it done somehow, because these guys are our friends. Anyway, if you guys have questions, we'd be happy to entertain them. Otherwise, back to work.
Thank you, Maureen. One question for the outer island growers: are they going to be able to get any of this clean tea
Sure.
cuttings?
Sure. We'll take them to state ag, and they will not have been in soil. So we'll unpot them, get them,
Okay.
we'll get them checked. We'll get them checked, because that's what we do with our papaya plants. They basically come looking for ants.
Okay. And then how much material can a grower get from you folks?
We don't know how much we can make. It's on this. Since May, when Myron called me up, "Hey, Maureen, you looking for another project?" "No, what?" So, it hasn't been that long.
Do you plan to expand your stock material or?
Of course, yeah. That's what we're doing now. We're not giving anything away. We're just cutting and cutting, and the microprops, as Mike said, they did tissue cultures, and you can get, he said he had one little 4-inch box with 1,000 plants in it. He's never gotten that again, and I hope we don't get that again. But
Okay.
yeah, we have been told by John Kunasaki, who's a very good tissue culture fellow, that you shouldn't grow them too quickly, because you don't want to get off types. Like, think
Right.
about bananas and some other crops, you get big ones, little ones, weird stuff. So
Okay.
we're plugging along.
All right. Well, thank you very much for your work on that.
You're welcome. It's fun.
Okay, great.